All right, guys. Well, welcome. AP Psych. I am doing your Sensation and Perception Week 6. We're doing the ear and the eye today, so we're going to get straight to it. First and foremost, my thoughts are with you guys. I'm really bummed I'm not seeing you till Monday. I was hoping, we could do this together. But it's not a big deal because some of you really don't have power. Please know that you are with me in my thoughts at all times. I'm really kind of worried about you guys. So, I can't fix power lines. If I could, I would. But I can teach this. Now, keep in mind, if you are out of power, this is not what you should be doing. If you're out of power, you should be focusing on things you can handle. Like food, water, safety. If you will have power, though, I'm expecting you to be doing these lessons as we go. Yesterday, I did post the schedule to Instagram and to Twitter, so please make sure you look at that. Test on Tuesday, on week 5, week 6 test on Friday. We gotta just get going here because we're 7 days behind. But if you're with power, you should be working. If you are without power, my thoughts are with you. I'm so sorry. Let me know if there's anything I can do to help. So, what I can do is teach you about the brain. We already did the brain. All right, so let's begin. So, here we go. I like this stuff. I mean, it's biology round 2, but I like it. So, you do need your crayons or colored pencils or whatever you have. Where I write is where you should be writing as well, because we have a lot of stuff that we're going to be putting down here on paper. So here is your pinna, and it funnels sound waves into your auditory canal. Okay, for common language, we call it your earlobe. Okay, then you have your auditory canal, which common way is ear canal, of course. And then you have your, I'm going to put it right here, tympanic membrane, which is your eardrum. And it really does kind of work like that. So when we talk about everything from your pinna to your eardrum, we call this your outer ear. Now your outer ear is made up of your pinna, auditory canal, and your tympanic membrane. Okay? And your outer ear is processing sound waves. Okay? So I'm going to take my colored pencil and my crayon. I'm going to use blue for now, and I'm going to color in my pinna. I hope you are writing where I am writing, because we are really going to fill in this whole sheet of paper. So my pinna is part of my outer ear, then my auditory canal, and then all the way to my tympanic membrane. There you go. So your outer ear is everything in blue, and then of course we have it explained down here. So I'm just going to highlight that with my color crayon, and there you go. So here's your outer ear. Then you have your middle ear, which I'm just going to start with your tympanic membrane. Then we have something called your ocelles, which is made up of your anvil, hammer, and stirrup. These are these little bones right here. They are the smallest bones in your entire body. The big term for all three of them is your oscilles. The smaller term for each individual one is the anvil, the hammer, and the stirrup. Okay, and these are the smallest bones in your body, and then they connect to what we call the oval window. Okay, so the oval window connects it to this weird, funky little thing. For me, when we call this whole thing from the tympanic membrane all the way down to your oval window, we call this your middle ear. Okay, so your middle ear is made up of your tympanic membrane. There's always an overlap, guys. Nothing is truly unique. So it has to have an overlap, so that's why the tympanic membrane is at the very end of your outer ear and the very beginning of your middle ear. Then we have our oscilles. Our oscilles are your anvil, hammer, and stirrup. And stirrup. And then you have, of course, your oval window. Alright, so with a color, for me, I'm going to do yellow. You have overlapping, of course, of the tympanic membrane, then into your oscilles, which is broken down into three major regions, your anvil, your hammer, and your stirrup, and then, of course, your oval window. So I am going to just highlight my middle ear right here, and then we're good to go. So far, sound waves travel from, and we're going to write physical. I'm going to explain why here in a second. Okay, so when we talk about the sound waves, okay, we transition to this whole idea that our sound waves are going through our ear. See, look, who knew you were going to get an earshot today? Okay, so it goes into our pinna, through our auditory canal, into our tympanic membrane. Now, if you stick your finger deep enough, I don't really suggest this, by the way, you can touch your tympanic membrane. You may have hit it with a Q-tip if you've ever used one of those. Okay, that's all your outer ear. If you can hit it with a Q-tip, it's got to be your outer ear. Okay, so sound waves, literally sound waves, as you're hearing my voice come through, are being funneled into your pinna, through your auditory canal, and hitting your, as I'm like pointing to your little picture, okay? So they're funneled into your pinna, all the way down your auditory canal, and hitting your tympanic membrane. The sound waves are making your tympanic membrane vibrate back and forth. Okay, so as the sound waves hit your tympanic membrane, it starts moving the oscilles, which are little bones. So every time your tympanic membrane moves, it moves the bones. So we go from a we go from a visual component, we go from a sound waves into a physical movement once we hit the middle ear, which is really kind of cool. So it's that whole movement with the tympanic membrane vibrating from sound waves. Okay, so the pinna to the auditory canal to your tympanic membrane, then your tympanic membrane to your oscilles, then to your oval window. So your oval window brings us into our inner ear. Now our inner ear is of course going to start with the cochlea. So this is your cochlea. Now it is filled with mucus and is where transduction occurs on the basilar membrane. Okay, so what is your cochlea? So the cochlea is a snail-looking thingy that we have right here. Now we're going to make a couple adjustments because we're more sophisticated than this shows us. So inside your cochlea, you're going to draw a little lining, as I'm doing right now, and it's just going to go to here. And then on the inside again, we're going to draw the little lining. So on your lining, you're just going to draw little cilia. And we're going to, of course, represent that by little sticks. There we go. All right, so that is your cochlea. This, right along the lining of it, is called the basilar membrane. And this is where transduction occurs. Now, what is transduction? Transduction is what converts external stimuli like sound waves into a neural impulse that our brain can actually process. So if I cut off the skull cap of your brain and start shouting at your brain is going to be able to process the sounds because that's not how the brain works. You have to take your external stimulus, which is my voice right now. It has to go through this whole process, converted into what will be a liquid movement in a second. And then here at the basilar membrane, this external stimulus is converted into a neural impulse. And that neural impulse will travel down the auditory canal to auditory cord, auditory nerve. It's the nerve. To your brain, and that's how we hear things. So, in your basilar membrane, we are, which is inside your cochlea, is what is actually processing this information. Okay, now you also see that we have this thing right here. This is your semicircular canals. Okay, and this is responsible for balance. Okay, so let's color in our inner ear. Okay, and I'm also going to write inner ear. In your inner ear is everything from your oval window to your nerve. Okay, so I'm going to quickly color in my semicircular canals because technically it is in your ear, in your inner ear. However, please keep in mind that it doesn't actually help you with hearing. It's just to help keep you balanced and keep you on your feet. Of course, here's your cochlea, which we're going to draw a little bit more detail here in a second, which is why I have all this room down here. So your inner ear, which you're going to write down here, inner ear, is the oval window to the cochlea, which is your basilar membrane. And this is where transduction occurs. Then goes to your auditory nerve. Then to your temporal lobe. And then your auditory cortex. Okay, and for me it was pink. Almost did it with yellow, that would have been bad. For me it's pink. Okay, oh, I'm so sorry. And this is going to be liquid. Why liquid? Well, let me show you. So I'm going to draw a nice little box right here. And this is inside your cochlea. Okay, so inside your cochlea, you have your basilar membrane. Okay, then of course you have your cilia. And in here you have mucus. Okay, so inside your cochlea you have your basilar membrane. Okay, inside, on top of it you have your cilia. And your whole cochlea is literally a mucus-filled little snail-looking thing, which is pretty cool. So every time your cilia moves, it creates a neural impulse, which is transduction. Okay, so let's draw it. So our little cilia are sitting in a cochlea filled with mucus. So we're going to make our mucus green, obviously. Okay? And then we have our basilar membrane. And every time the basilar membrane, every time a cilia moves, it creates a neural impulse. And that neural impulse is causing transduction. So everything you're hearing right now inside your cochlea, every time the cilia moves one way or the other, it creates a neural impulse, which is so cool. So what happens is, when we're talking about your basilar membrane, so we have our little diagram, okay? So as the physical sound is coming through your ear, now you can hear me differently if you curl your pinna in and out. I am going to sound a little bit differently. If you do that while I'm talking to you, close it up, funnel it in a little bit better, you're going to hear me a lot differently, and that's because the pinna's job is just to funnel sound into your auditory canal, and then once it's in your auditory canal, it's going to go and hit your tympanic membrane, and that's going to take the physical sound waves and make a physical movement. So your tympanic membrane is vibrating every time you hear my voice. Then that tympanic membrane is going to keep shaking until it hits your oscilles, which is on the other side of that tympanic membrane, and so that vibration is going to create a physical movement. And then once you get to that physical movement, it goes all the way through your oscilles. It's then going to push and pull on your oval window. Once it pushes and pulls, it takes the oval window, which is holding back all the mucus, and it literally moves the mucus back and forth, and that mucus makes your cilia on your tympanic membrane make the neural impulse. It's transduction. Isn't that cool? I think it's cool. Anyway, so that's how you're hearing my voice. It goes from your pinna to your auditory canal to your tympanic membrane to your oscilles to the oval window inside your cochlea to the basilar membrane where neural impulse or transduction is occurring. Then you are going to have transduction occur and then all the way out to your auditory nerve. How cool. All right, so to make it a little easier, let's write it all down how transduction occurs, because on your test next Friday, you will have to know the correct order of how a neural impulse is converted in how your ear creates transduction. So you have the pinna, then goes to your auditory canal, then to your tympanic membrane, all of this, of course, is in your outer ear. And then you hit the oscilles, which are those three little bones, the anvil, hammer, and stirrup. Then you hit the oval window. Of course, all of this is the middle ear. Then you have the cochlea. Then the basilar membrane. This is where transduction occurs, and you need to know that because transduction is a very big deal. Then you have the auditory nerve. Then you have the temporal lobe. Then you have your auditory cortex. All right, and of course, all this is in your inner ear. All right, so of course, our outer ear is blue, so I'm going to just quickly color this in, just have a nicely color coordinated here. Now, keep in mind your tympanic membrane is, of course, both your end of your outer and the beginning of your middle. And of course, yellow is my middle ear. And then I have pink for my inner ear. All right, guys, that is it for the ear, which which is pretty awesome. So it goes pinna, auditory canal, tympanic membrane, oscilles, anvil, hammer, stirrup, then to your oval window, then to your cochlea, then to your basilar membrane, where transduction occurs, then to your auditory nerve, then to your temporal lobe, and then to your auditory cortex. All right, there you go. So let's flip it over and let's start the eye. I like the eye as well. So right where I am telling you, actually, let's just make a quick little note about transduction before we move on, because transduction is really what this whole week is about. So transduction is the conversion of an external stimulus, in this case, of course, it's sound, into a neural impulse. Okay, so transduction. This is really what everything's all about this week, transduction. All right, join me on the other side. So flip it on over. All right, so first we're not going to use all the lines. This is a little more sophisticated than we need, but we are going to use some of them. So it goes cornea, which is right here. Now your cornea is a clear, glass-like layer over your eyes. Over your eye, which people usually consider iris and pupil. It is the only part of your eye you can touch. All right, so your cornea is what you touch when you put contacts on. It's this outer part right here. So you're going to take, I'm going to use blue. Okay, it's this part right here. It's this outer part. This is all your cornea. It allows you to see through and protects your eye. Your cornea. Okay, so your cornea is right there. Then you have your pupil. Your pupil is a hole in your iris that allows light to enter your eye. Parentheses, black spot. Okay, so your pupil really isn't actually anything. It's a hole. That's what it is. So when people talk about your pupil, it's actually not a thing. It's the absence of a thing. So if you look at mine, it's that black center of your eye. It's not a thing. It's actually the lack of something, which is pretty cool. So that's your pupil. I'm going to leave that blank because it doesn't actually connect to anything. So I'm going to leave that alone. So, oh, you don't care. So your next one is your iris. And your iris is the colored part of your eye. That is made up of muscles that constricts or dilates your eye, controls the amount of light entering. So for me, this person is going to have pretty green eyes. So, because I already used blue, and mine are blue, so I'm going to use green, even though no one in nature has green eyes that look like that, but it's fine. Your iris is around and surrounds your pupil, and they're just little muscles. That's it. Okay, that's all it does, is that it's just little muscles and it opens up your eye, your pupil, which is just a hole, or it constricts it and makes it really, really small. So it's either make it really, really big, so that would be a large pupil. It means a lot of light's coming through, which makes it really hard to see outside specifically. And then, or it constricts, which makes it smaller, and once it makes it smaller, it means less light's coming through, which means it's really bright outside. So you have the iris, and then right behind the iris, you have the lens. Now this focuses and flips the image upside down to make it easier for the retina to process. There you go. So the lens focuses and flips the image. So when you're looking, it flips the image upside down completely, which is really crazy. It just helps your brain process better. Isn't that so strange? And that's exactly what it does. And it's located right behind your pupil. So when you're looking at black, you're actually looking at your lens, which is pretty cool. If you go to your optometrist or the guy or the person who prescribes you glasses, they're the ones looking at your lens, and through the lens they can see your retina, which we're about to talk about here. I'm just gonna do a better job about connecting my colors, just so there's no confusion about what is what. There's your cornea. All right, so next is your gratuitous humor. And this supports the structure of the eye. There you go. It's like a jelly. It's like a clear jelly. If you've ever dissected a cow's eye or anything, it's like a little goop that comes out. And that's what it is. So it's just like jelly. It just keeps the eye and gives it its structure, which is pretty cool. All right, so next we have your sclera. We're going to go way up here. Your sclera. It is the eye muscle outside of your eye, of your eyeball, that makes your eye move. That moves your eye. There you go. So with your sclera, okay, now I want you to do this, but I don't want you to hurt yourself, okay? Go. See how I'm moving my eyes back and forth? Do that. You could feel like a muscle moving in your eye. That's your sclera working. Isn't that cool? Anyway, getting back to it. All right, so your sclera is on the outside, and for me, I am going to make that yellow. And it's just on the outside of your eye, and it is just helping it move and adjust. Technically, your sclera is part of this. These muscles here are part of your sclera, and it just controls where your eye is going. It's on the outside of your eye. There you go. All right, so next is your retina. Now, your retina is the most important part of your eye. This is where transduction occurs. Your retina. Now, your retina, this is where transduction occurs. Light is turned into a neural impulse. Okay? So, for me, your retina is going to be red. Your retina is on the inside of your eye, of course. And the closer it is to the back, the thicker it is. All right, and then right here, you have your fova. Okay, and your fova is actually right here. So I'm going to take a black crayon and I'm going to color in a very big X. Okay, so your fova, also known as focal point, this is where the image hits the retina, and this affects the image's clarity. Okay, so your fova is just the specific spot the light comes through and touches. Now, this is your optic nerve over here. And this sends neural impulses to the occipital lobe to be processed by the visual cortex. So this is just carrying it out. Okay, so I'm going to make this light green. And it's located right here. Now there's one more thing you need to know. So I'm going to take this really ugly, I guess it's not ugly, it's purple. Right here, I'm going to draw a nice big circle right in front of my optic nerve. This is my blind spot. Anything, any image that hits the blind spot is not seen. Okay, so what is happening here is that your light, anything that goes right onto your optic nerve, means it didn't touch your retina. If it didn't touch your retina, then it cannot process the information. Okay, so that's what the blind spot is, which we're going to get into more discussion a little bit later. All right, so what happens is I'm going to take yellow and I'm going to draw, I guess I should use a different yellow. I'll grab a nice pretty light yellow. So as light enters your eye, it's being filtered into your eye through your cornea, which is protecting your eye, into your pupil, which is of course nothing, but is surrounded by your iris, which is the colored part of your eye. The light goes through your lens, it is sharpened, and it is focused, and the image is actually flipped. Then the light goes all the way through your vitreous humor and hits your focal point. Once it hits your focal point, then it's on your retina. So the focal fova is located on your retina. Once the light hits your retina, then transduction starts occurring. So what is actually happening? So on the very bottom, you should have space. Remember, I told you to write where I write. So let's talk about it. So this is your retina. Okay, so light hits retina. Okay, so we're just going to draw light hitting it. Okay, so this is on your focal point, of course. Then we have 3 different types of cells that are processing it. So we're just going to draw them out together. In case you can't see, I'm alternating rods and cones. Alright, and now I'm just going to connect it to my little endpoints here. Alright, so what we have here is we have 3 different parts. So, first thing, light hits the retina and is then processed by what we call ganglion cells. Ganglion cells are the first layer of your retina. So the first layer is your ganglion cells. Then your second layer is what we call your bipolar cells. Your bipolar cells take the initial breakdown that the ganglion cells did of light and then processes them. And then your third one is your rods and cones. Your rods and cones is where we start seeing color. So your rods are looking at black and white images only, while your cones, see, cones is for color. Okay, and then the fourth layer is transduction occurs. So what happens is that your ganglion cells, which I'm going to underline with purple because they go there, then your bipolar cells, which I'm going to color in green, are located right here, and they are processing the information that is coming from the ganglion, and it goes to your rods and cones. And then with your rods and cones, they go here. It's all this stuff over here. And then finally, you have transduction, and that's going to happen back here. So once it goes through your rods and cones, then transduction occurs. So, the light hits the retina. The retina breaks it down immediately by ganglion cells, then bipolar cells, then rods versus cones. Once its rods and cones have processed, then actual transduction has occurred, and that is where the actual retina is. Isn't that cool? It's so cool. So, everything's color coordinated. However, we need to make sure we know the order of how the eye fires. So, we're going to write it way over here. Write it as best you can, as in smaller space as possible. So, your cornea goes through the cornea first, then to your pupil, then to your iris, then through the lens, then hits your fova, also known as your focal point, then goes to your retina. And we know the retina is where transduction occurs, but it goes through your ganglion, then your bipolar, then your rods and cones. Then it goes through your optic nerve, then to your occipital lobe, then to your visual cortex. There you go. So, how does light go through the eye? So light goes through the cornea to the pupil, which doesn't actually exist. The pupil is surrounded by the iris, then the light goes through the lens, which is then flipped upside down and then hits your focal point or your fova. Your fova is located on your retina, and your retina has your ganglion to your bipolar, then your rods and cones, and then it goes to your optic nerve, which is right here. And then from your optic nerve to your occipital lobe, then to your visual cortex. There you go, guys. So everything's there, everything you need. Yeah. So I hope this video finds you in a place of comfort and air conditioning in your house. But please know you are in my thoughts, and I am going to keep making videos. I'd much rather you with me, though. Anyway, my thoughts are with you. If you need anything, please, 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 please reach out. All right, bye, guys.